Welcome to Lecture 17, Transcription, Translation, and the Structure of the Gene. So just a reminder again of the flow of information in the cell. The central dogma posits that the flow of information from nucleic acids, either DNA to RNA, enters into protein through translation. So transcription takes DNA and processes the information into RNA, and it's translation that processes nucleic acid information in the form of RNA into protein. So here we have a sequence ATG, TCC, so on and so forth. Transcription generates a nucleic acid sequence in mRNA form, AUG, UCC, so on and so forth. And then the tRNA molecules recognize the triplet codon sequence, and the tRNA molecules are also tethered to amino acid sequences, providing in this particular case a chain of amino acids. All right. One thing to remind you about is that the central dogma posits that information flows from nucleic acids into proteins, but not proteins into nucleic acids. So it doesn't specifically say that DNA to RNA to protein, because there are ways in which RNA can actually be reverse transcribed into DNA. But generally, the flow of information in the cell occurs according to this flow. So DNA, transcription, RNA, and then translation. So just a reminder, you've seen this slide a couple times. RNA is a little bit different than DNA. All right, one of the critical things is that the 2' hydrogen in DNA, all right, which gives it the name deoxyribose, the 2' actually is a hydroxyl, an OH there, all right. And another large difference, instead of thymine, one of the bases is replaced with uracil. But what is similar to our DNA is that it's a 3' end that grows, and it's that 3' hydroxyl that's critical. So here we have a stretch of DNA sequence. Here's the 5', here's the 3', all right, single-stranded, and we have an RNA molecule that's being read and is extending, all right, reading this template and extending at the 3' end. So here we have a nucleotide of RNA with the phosphate about ready to be joined with this hydroxyl. So there are 3 major flavors of RNA in the cell. There's lots of different other kinds that we won't get too much into. The one that you're probably most familiar with is mRNA, or messenger RNA, or message RNA. It codes for proteins, and so the RNA molecule that I showed in this slide here would be an mRNA because it has a coding capacity. And it is synthesized by RNA polymerase II in eukaryotes. There's another very important RNA called rRNA, or ribosomal RNA. And ribosomal RNA basically contributes to the structure of the ribosome. And the ribosome is the molecular machine that translates mRNA into protein. And so it forms a very large structure of the ribosome, and ribosomal RNA is an important part of that structure in addition to other proteins inside the ribosome. Ribosomal RNA is synthesized by RNA polymerase I in eukaryotes. And then in addition to that, there's also tRNA, a transfer RNA, and that I've already shown you briefly carries amino acids to the ribosome and mediates the translation of mRNA into protein sequence. And tRNA is synthesized by RNA polymerase III. So what's interesting is you have these 3 different RNAs, but they all contribute to the production of proteins in very different ways. The mRNA codes for the proteins. The ribosomal RNA plays a critical role in forming the structure of the ribosome, and the tRNA is that link between nucleic acid world, because it recognizes the codons, and amino acid world, because it's tethered to a specific amino acid for a specific codon. So continuing, in order for RNA to be transcribed, you can see that, of course, DNA, which is double-stranded, has to be opened up to make something called a transcription bubble. So this particular stretch of DNA, which is double-stranded, opens up in this particular way, and here it's the 3' end that is extending into that transcription bubble. All right. So if the RNA is growing at the 3' end, what direction is the DNA transcribed? All right. Well, in this particular case, this particular molecule below is the template, right? I mean, it's two stretches of DNA, but it's the bottom one that's the template. And that's the template because this is the 3' end, so this is the 5' end. Okay, so the direction of the new RNA strand is extending at the 3' end, but reads the DNA template from 3' to 5' because it has to extend the 3' end. Now, you might be saying to yourself, wait a minute, it looks like only one strand or two strands, like how many strands, like what's going on with the strands of the DNA? Because there's a top and a bottom, all right? It's important to understand that different strands can code for different genes. So what I want you to recognize here, this section here is called gene 1, right? And in this particular case, gene 1 is transcribed in this direction. So the 3' end reads and extends here, reading the template in that direction. Now you might be saying like, well, what about this part of gene 1, right? Because this is the reverse complement. Gene 1 is only transcribed from this bottom dark blue strand. It's not transcribed from this top strand. However, gene 2, right, which is again shown in dark blue, is also transcribed from that top strand, right? And so again, 5' to 3'. And you can see that the transcription, of course, is going in the opposite direction as a transcription from gene 1. And then you have gene 3 that happens to also be transcribed in this direction. So what determines the coding sequence, of course, relies on the direction of transcription, because this particular gene, in principle, I suppose, could be transcribed in both directions, but in this diagram, it's only transcribed in one direction. This one's only transcribed in one direction, and this one's only transcribed in one direction. So if you were to kind of look at it at the nucleotide level, here we have gene 1, all right, and it would be transcribed. In this particular case, it's transcribed in this direction. So it's not the same as gene 1 and gene 2 from the previous image. Just think of them separately. Here we have a particular gene. It opens up. Here's the transcription bubble. Here's the enzyme called RNA polymerase. And RNA polymerase is the enzyme. It's actually a machine, in fact, that extends the 3' end. So here we have a 3' end that's growing in this direction here, reading off of the bottom strand here. But in this particular case, for gene 2, it's the top strand that's being read, and then the transcription is going into this direction. So the synthesis of the 3' end proceeds through this unwound, what is called a transcription bubble, by the molecular machine RNA polymerase. Okay, so this is worth thinking about. Is there then a leading and lagging strand for transcription? Remember, you have basically this bubble, which we call the transcription bubble. Is there a leading and lagging strand? Right? No. Leading and lagging strand, so the answer here is B, no. Leading and lagging strand only apply to DNA replication. All right? Because with transcription, when a gene is being transcribed, it's being transcribed at a particular moment by an RNA polymerase in one direction only. All right, it won't be transcribed in the other direction, both simultaneously. All right, so the answer here is no, there is not a leading and lagging strand. There's only one strand for transcription that's being synthesized at a given moment, and it wouldn't be designated either of these. Okay, so we're going to talk about the ways in which RNA transcription and translation work. And we're going to start off talking about prokaryotes, and then we're going to switch to eukaryotes. Now, a critical feature of prokaryotes, and I'll mention this again and again and again, is there's no specific nucleus. Okay, there is a genome, of course, but because there's no nucleus, what this means is that transcription, here that we have a growing strand of RNA, and translation, here we have ribosomes translating the growing strand of RNA to make amino acids. those two processes are very tightly coupled. So with regard to transcription, the RNA polymerase is something called a holoenzyme. A holoenzyme is a complex of 5 different subunits. In the case of prokaryotes, it consists of 2 alpha subunits, 2 of these beta subunits. And then it consists of this other factor, which is called the sigma factor. And the sigma factor plays a very important role in pulling the polymerase machine here to the DNA sequence. So it basically identifies the DNA sequence with assistance of these little sigma factors. You'll notice here I have these numbers here, -10 and -35. That is part of the sequence of the DNA sequence that the sigma factor binds to. And -10 and -35 refer to a relative position relative to the place where transcription actually initiates. And I'll show that more clearly in the next picture. So this is what the structure of DNA sequences that drive transcription look like in a prokaryote. So there's a structure called a promoter, all right? And then of course here's the coding sequence of the gene here, and this is being transcribed. And the transcription is initiated through the binding of the RNA polymerase holoenzyme to the promoter, all right? But the transcription directly starts at a position called +1. The +1 position is the first nucleotide that's transcribed. Okay? And so you can see with this growing RNA molecule here, this is going to be the 3' end that extends. The first nucleotide is shown at this +1 position. All right? So this will refer to the ATG, which is the start codon, which would be an AUG in the RNA sequence. You can see the AUG in the RNA sequence right there. But what initiates at the +1 position, which is the first nucleotide that's transcribed by the holoenzyme, is that there are DNA sequences inside the promoter that allow those sigma factors to identify and drive transcription. And those important sequences tend to be at what is called the -10 position. So here's the 1 position, all right? So this will be -1, -2, -3, -4, -5, -6, -7, -8, -9, -10. So somewhere around -10. It's not always precise. You can see that there are some gaps. And then approximately -35. You can see it's between 15 and 17 base pairs away. From there, about there, if you line up promoters, which are the sequences of DNA that recruit the RNA polymerase, if you line up the promoter sequences of many, many, many different genes, you'll see a tendency for certain DNA sequences. A very common tendency here at this position is a sequence TATA. Now this is not universal, so for example this gene here has TATG instead. But there's a common motif of TATAAT, and then likewise there's a common motif here, excuse me, that those two cooperate to recruit those sigma factors. Now this is the sort of, this variation among different promoters, but this is a general observation that these particular sequences are critical sequences inside the promoter. Now it's the promoter that determines the direction of transcription. Okay, so what I have in this particular diagram here is I have a DNA strand here, and I have a DNA strand here. In this particular case, there's 1, 2, 3 genes, and each of those 3 genes generates 3 different transcripts. It's the promoter that determines the direction of transcription. So in this particular case, this promoter says transcribe going that way. And this promoter says transcribe going that way. And this promoter says transcribe going that way. So that's what I said before, is that the transcription will occur just in one strand, but it could be either strand. And that is going to be determined by how the promoter is positioned relative to the gene. If you flip the promoter around, if you inverted it, it would potentially change the direction of transcription. So once the RNA polymerase binds to it, then of course elongation must proceed. and that requires helicase activity, which leads to the unwinding of the DNA strand, and the RNA polymerase will extend down the length of the DNA as it unwinds. Now, one thing that often is thought is that, like by people just learning this, is that the way it works is sort of in the stepwise process. Was the RNA polymerase will land on the promoter, it will initiate transcription at the +1 position, and will go all the way to the end of the gene, and then another RNA polymerase will land, and then it will go all the way to the other end of the gene. But that's not how it works. All right, so here we have a DNA sequence, which is including the promoter. We have a transcription bubble. The RNA polymerase is moving down here. Eventually, it will get to a point where it stops, right? Because it's not going to transcribe the entire genome. But it isn't like the promoter waits for this RNA polymerase to get to the end before it fires off a new round of transcription. So it can continuously happen. So down in this scheme below here, what you can see is that this particular gene has 1, 2, 3, 4, 5 different RNA polymerase complexes that are transcribing the RNA. All right, and this one has just initiated, and this one's further downstream, and this one's further downstream. So all of this can happen simultaneously if there's a high level of expression for a gene. And you can see this particular pattern here. This is an electron micrograph, and this is the beginning of a transcribed gene, and this is sort of towards the end. And these growing sort of molecules that get longer and longer as you get further down the gene represent these growing RNA strands. So then you might be saying to yourself, okay, fine, I can understand how transcription is initiated, but how does it stop? All right. The book goes into a little bit more detail than I really care to go into here. So the primary mechanism that I'm going to discuss is basically the formation of a hairpin. All right. So RNA has a really interesting ability to fold back on itself. All right. So if you have an RNA molecule here, right, and this is the sequence, notice that there's a section of it in which this part, this part, this part, all is complementary in the opposite direction. All right. So this structure is called a hairpin, and this can form within any RNA molecule. And so oftentimes the transcription stop is encoded in the DNA to encode a hairpin in the RNA. Then once the hairpin of that forms, the polymerase will encounter a series of U's which cause it to sort of backtrack, and once it backtracks, it will identify the hairpin and it will release the RNA polymerase. The RNA polymerase will fall off and it will cause a transcription termination. So that's pretty much all I want to talk about the machinery of transcription in bacteria. What we're going to do is we're going to now highlight the differences between the machinery of transcription in bacteria with the machinery of transcription in eukaryotes. And there are 4 very big differences between eukaryotic and prokaryotic genes and gene expression. We're going to walk through those now. Okay, I mentioned this already once before, but I'll mention it again. In eukaryotes, translation occurs outside of the nucleus. So here we have in DNA, a prokaryote. The DNA, is a circular molecule typically, and the transcription is occurring. But as that growing strand of RNA is being transcribed off of the DNA, it can be immediately translated because there's no partition enforced by the nucleus. However, in the case of eukaryotes, there's a nucleus. Okay, and so what that means is that all of transcription has to be finished. The RNA molecule has to be processed and then transported out of the nucleus, and then the transcript can be translated. All right. So you might be saying, well, that might not be that important, but it's extremely important because it plays an important role in determining how gene regulation occurs, because these are critical steps between the generation of the RNA and the generation of the protein that don't occur in this particular case, in the case of prokaryotes. Okay, another big difference is complex chromatin. All right, we talked about the 30 nanometer fiber. All right, DNA in eukaryotes is wrapped around histones. And what that means is that when the histones are compacted amongst each other, that renders the DNA sequences embedded in that sort of compacted structure inaccessible to RNA polymerase. So in order for genes to be expressed, the histones need to be basically wrestled with, to be moved away to allow the RNA polymerase complex to land on gene sequences and then generate transcripts. Another difference is that the RNA polymerase complex is much more complicated. All right. So before I showed, 2 alpha factors, 2 beta factors, and a sigma factor. There was another little factor that I didn't want to get too much into. But RNA polymerase 2, the complex in eukaryotes, contains 12 different subunits. The other thing that is somewhat similar is that they still bind DNA with assistance of transcription factors, but they're not exactly like the sigma factor. An additional difference, and this is very important, is that the structure of genes is very different between eukaryotes and prokaryotes. All right. So similarly, there is a promoter. All right. So this is a structure of a gene here. All right. And transcription will start at the promoter. So that's something similar between eukaryotes and prokaryotes. And oftentimes you'll see a TA sequence. But what there are, after transcription begins, there are sequences at the +1 position, all right, that correspond, of course, to exons, but then there are other sequences called introns, all right? So this entire sequence here is going to be transcribed from the +1 position all the way to the transcription start site, from here to here. But the introns are spliced out of the mRNA. So what you end up with is, from this structure where you initiate transcription here, I want you to notice something. The start codon is not at the +1 position. The start codon is a little downstream. All right, and that's because the start codon has nothing to do with transcription. The start codon has everything to do with translation. Likewise, the stop codon has nothing to do with transcription stop. The transcription stop is down here. So when this RNA molecule is processed, what you end up with is a sequence that is at the very beginning, all right, which corresponds to the RNA sequence prior to the start codon. This is called the 5' UTR, and UTR stands for untranslated region. So this little blue section here is untranslated. But it's still exon, and the reason it's called exon is because we define exons as the part of the mature mRNA that is not spliced out. So exons may not code for protein. Another part of this mRNA transcript that doesn't code for protein is called the 3' UTR. Here's the translation stop of the sequence, all right? But downstream of that, because translation is stopped, this is RNA sequence that doesn't get translated. So we call that the untranslated region, or the UTR. So initiation also occurs at the promoter. It's mediated by factors called general transcription factors that pull core proteins in the core polymerase to the TATA box. Okay, so there's that TATA sequence. Similar to prokaryotes, is about 30 base pairs away from the start site of transcription. A protein, in this particular protein, TBP, stands for TATA binding protein, binds to the TATA sequence, and then TF2D, which stands for transcription factor 2D, in turn binds to the TATA binding protein and then recruits this large set of other transcription factors. TF stands for transcription factor, so TF2F, TF2H. You don't need to worry about the names of those particular transcription factors, but altogether they then pull the RNA polymerase to the site of transcription. Now, a critical thing that happens to the RNA polymerase, so the RNA polymerase complex, that 12 subunit complex, which I showed a kind of a complicated crystal structure for, is shown in purple here. the way in which it initiates transcription has to be tightly regulated. And the way it's regulated is through what is called phosphorylation of the protein sequence. So phosphate groups here, you can see them littered along, are cover what is called the carboxyl tail domain, right? So the carboxyl tail domain is the very, it's one of the it's the carboxyl end of one of the protein sequences that gets modified, right? And it gets phosphorylated. And it's the phosphorylation status that will govern the way in which the RNA polymerase processes down the length of the gene. So, as I said before, it also occurs at promoters. Here we have a transcription start site for many, many different genes. And then you can see that there's a tendency for TA sequence to be in front of this at about 25 to 30 nucleotides, but you can see that's not always going to be the case, but there's a tendency for it. So it's worth thinking about what genes look like in humans, considering what we were just talking about. So I'll just walk through some numbers here. The median, I'll just stick with the median, but you can look at the mean as well. The typical size of an exon inside a gene is not that long, right? It's only 122 base pairs. And a typical gene has about 7 exons. The introns are much larger, which is curious, right? Why so long? All right, they're about 1,000 base pairs each. And then that 5' and 3' untranslated regions are 240 and 400 base pairs about. And then the length of the coding sequence is about 1,100 base pairs, which, will code for 367 amino acids. But the entire gene is 14 kb or 14,000 base pairs. Excuse me, kb is 1,000 base pairs, so 14,000 base pairs. So 14,000 base pairs of all this extra stuff, which only encodes for 1,100 base pairs, which is a little bit interesting. Okay, so those are some differences. We're going to keep on trucking. A critical difference, and I've alluded to it already, is that RNAs are processed in eukaryotes very differently than they are processed in prokaryotes. So we're going to walk through what I mean by mRNA processing. Okay, the mRNA in eukaryotes is modified in 3 different ways. One of those modifications is known as capping. All right, so here, this is the very first nucleotide of a growing RNA molecule, the second nucleotide here. And so, here's the, excuse me, no, this is the entire structure. Okay, so this is going to be some nucleotides, and there's a particular sequence that is like almost reverse, all right, to the direction of our typical RNA molecule. So notice we got a 5' here, a 5' here, and we get the phosphate groups. Okay. What's kind of curious about this, though, is you can see that there's a phosphate group here that joins another 5'. All right. So we have a 5' to 5' connection, which is really weird. All right. And this is a methylated cap. And so this entire structure, these are guanosines. All right, is what is called a 7-methylguanosine cap, right? Which is a very strange structure that would be introduced directly to the beginning of the growing RNA molecule. Now, what is the purpose of this? Remember, the RNA molecule has to exit the nucleus and go into the cytoplasm. This is basically a critical event that allows a RNA molecule to be considered fully processed. It's sort of like a flag in a way that shows like, okay, the right thing has been done, and it's critical for initiating translation in the cytoplasm and stability. So if an mRNA is not capped for whatever reason, like a mistake is made, it will be broken down. It won't ever leave the nucleus. Okay, in addition to that, we also have this splicing reaction. So this is an RNA molecule. Here's exon 1, here's exon 2. And remember that critical difference between RNAs and DNAs, or riboses and deoxyriboses, is that 2' carbon there in RNA is an OH group, all right, which is fairly reactive. All right. So what that means is that that reactive group can do stuff in the RNA molecule itself. And one of those things that it can do is splice out intronic sequences that do not typically code for anything. Okay, so what you have here is you have this 2' hydroxyl here. It can attack one of the phosphate groups, and by doing so, it can break basically the strand of RNA. Now, once it breaks that strand of RNA, there's going to be an OH at this position here, and that OH can then attack this phosphate group, and by doing so, you end up basically splicing these two exons together in this particular configuration, and you get this X-sized RNA molecule that's basically sort of circular, and it's called a lariat. Okay, and so that's an interesting feature, and you might be wondering why the heck cells even do this, and we'll propose a reason maybe why in a moment. Okay, now there are RNA sequences that play critical roles in that splicing reaction. Okay, so this is a typical, RNA sequence that one might see at an intron. Here's a 5' exon, here's a 3' exon, so the red is the exons, and then in brown here are introns. And what I show here are the frequencies of nucleotides at these particular positions when compared across many genes. So basically 100% of the time you have a G and a U at this position within an intron, and an A and a G at this position within the intron. And then there's going to be an A, which is going to be 100% present, but of course it kind of can vary a little bit at the exact position. This is that branch point that initiates the splicing reaction off of this position right here. Now within the exon there is a little less conservation. You might see a G that's commonly there, you might see a G that's commonly there, but there's a particular motif within the intron that initiates this reaction. But it's not occurring by itself, okay? There is a whole set of little tiny RNAs that I'm not going to go into the details of naming, but there's a whole molecular machine that processes that. So it's not like the RNA just splices itself completely on its own. There are little helpers that help that process, and those are other little RNAs, which is interesting. So remember I said that there are mRNAs, tRNAs, and ribosomal RNAs. Well, these are some other RNAs that help enable splicing. Okay, so that is the splicing reaction that happens to mRNA. That's one of the processing. And then the third form of processing is called poly-A tail addition, or often usually called polyadenylation. Again, these are sort of modifications that happen on a synthesized RNA strand that are critical for stability and for export from the nucleus. So I won't get into the exact nature, like in the transcriptional stop process in prokaryotes, I discussed the formation of a hairpin. Oftentimes, in this particular case here, we have a growing RNA strand, and this is the 3' end here, and this is the 5' cap. There are particular sequences that are recognized to initiate transcription stopping. All right, but I won't get more into the details than that. Okay, but those sequences get recognized, and then the RNA structure can be cleaved, which can enable transcription stop. And once that cleavage reaction occurs, a stretch of A's are added by an enzyme called poly-A polymerase, and so that introduces a large poly-A tail. Okay, now oftentimes, and I remember when I learned this the first time, I had this sort of idea that you had an RNA molecule that got transcribed, and while it was still in the nucleus after transcription, then it got capped, then it got spliced, and then it got polyadenylated. And it kind of seemed like that all occurred after transcription. But the important thing is that that's not true. All right, all of this stuff is happening during transcription itself. Now remember I talked about how the RNA polymerase has this CTD, or the carboxyl terminal tail there, and that is highly phosphorylated. All right, so phosphate groups can be, and enzymes, other proteins do this. Well, those phosphate groups play critical role in recruiting the enzymes that do all of the stuff that I was just talking about. So at the very beginning, as transcription begins, all right, capping enzymes are recruited to the RNA polymerase. And so now these capping enzymes are recruited to the RNA polymerase, and they cap the transcript right away. All right, so capping happens right when transcription starts. In addition, splicing happens during transcription. Okay, so those phosphate groups also are able to recruit the RNA splicing machinery, which includes some of those little RNA molecules that facilitate this. And during transcription, these exons will get spliced and retained, and the introns will get spliced out. So that happens during transcription. And then finally, at the very end, during transcription, the poly A tail will be added by enzymes that are recruited by that phosphorylated CTD of the RNA polymerase complex. Now, we started off talking about, or we didn't start off, but I mentioned briefly, like, wait, why are there these exons and introns? It seems so weird. Like, what's the point of these introns? To be fair, it's not 100% fully understood. But there are some theories about this. Now, I talked about, mutation happening in DNA sequences during DNA replication. But it turns out mistakes can be made during transcription. So a newly synthesized RNA molecule is generated, but the wrong nucleotide is placed in there. Now, why would that be problematic? Well, that might be problematic if the transcript that's generated has a premature stop codon. A premature stop codon could produce a protein that is truncated. And that protein that is truncated could potentially be harmful. In fact, it could have a dominant poisonous effect. It's not maybe just like, oh, it doesn't do anything. It could actually be directly harmful to the cell. So there are systems in place that ensure that the RNAs that are generated are at least, not full of mistakes. Okay? And one of those sort of processes by which RNA are screened for the proper sequence is through something called nonsense-mediated decay. So by nonsense-mediated decay, I mean is that basically if a nonsense mutation occurs, I shouldn't maybe call it mutation, but a nonsense mistake occurs in the RNA transcript, the RNA can be targeted for degradation. All right, and that's why it's called nonsense-mediated decay, because nonsense mistakes will be errors that cause a premature stop. So I want you to look on the left here. Here we have a transcript. All right, it has exons in blue and introns in gray. All right, and this is the normal transcript. At somewhere early on in the transcript, there's a start codon, right? And then you move along the length, and then somewhere at the end is a stop codon. Keep in mind, there's not a stop codon at the very beginning because there's a 5' UTR here, and then there's not a stop codon, there's not a start codon at the very beginning because there's a 5' UTR here, and there's not a stop codon at the very end because there's a 3' UTR here. But these gray introns are spliced out. This is all well and good, but what is retained at those exon-exon junctions are proteins called the exon junction complex. So those are actually retained. Those aren't like, it's not just like naked RNA being placed into the cytoplasm. There's sequences, there's protein structures that actually retain. All right. Now, this is a proper RNA transcript. It's capped, it's adenylated. There are exon junction complexes here, and the start codon is in this position, and the stop codon in this position, and all well looks good. The ribosome will come along and translate it and displace those exon junction complexes and translate the protein. But let's suppose there's a mistake that's made. All right. So instead of the stop codon being sort of towards the end, there's a stop codon inside this exon, but then there's further potential coding exons. So this stop codon is a transcriptional error. All right, this is a mistake that occurred. And so what happens is there's a stop now that's retained. Now what I want you to notice here is that this means that there's a stop codon upstream of an exon junction complex. Here that's not the case, but there's a stop codon upstream of that. So when the ribosome recognizes the start codon and begins translation, it sees a stop codon, but then continues and recognizes that there's an exon junction complex downstream of it. That is a warning sign that this RNA molecule is somehow defective. So what this means is that this will then release, and then that RNA transcript will be degraded by that nonsense-mediated decay pathway. And a very fascinating thing and an important thing about splicing, again going to this question of like why are there splicing and why are there introns, why are there exons, is that a form of splicing called alternative splicing can establish a great amount of protein diversity. All right. So one of the types of alternative splicing that I'm talking about is a cassette exon. So you can have a particular, here's an exon, here's an exon, here's an exon. Under some conditions, you can have a splicing reaction that joins this exon to this exon, and you produce this transcript that will be translated. But under other circumstances, the splicing reaction can splice this exon to this exon, like in this fashion. And so now you have a single gene that, depending on how it gets spliced, can produce transcripts that look like this, or transcripts that look like that. Okay, you can also have mutually exclusive exons. So in this particular one condition, we have splicing of this one, the pink one, and then to this gray one, and that will produce this type of transcript. Alternately, we can have this one skipped and instead favor this one to be spliced in, and you produce this one. So you can see that, because that the coding sequence inside these different exons, will code for different proteins, you can see how one gene can code for multiple proteins this way. And this highlights a really fascinating example of a gene called DSCAM, which has a structure that looks like this. All right, there are these sort of exons inside this gene sequence that are designated as A exons or B exons or C exons or D exons. So these are normal exons. So exon 1, exon 2, exon 3, they all get spliced in. But among the A exons, one of those is randomly spliced in and the others are randomly spliced out. Then you have another exon, and then you have among the B exons, ones of those will get randomly spliced in, and then the retaining will be spliced out. So forth for here, so forth for here. And so in this particular mRNA, you have, based on splicing, one of the A exons designated 8, one of the B exons designated 24, one of the C exons designated 16, and one of the two D exons here designated D2. Now what this means is you can imagine that there's a lot of different combinations. In fact, there are more than 38,000 possibilities from a single gene based on how it's spliced. And the stunning thing about this is that number of possible gene splice forms from this single gene, from this thing called gene called DSCAM, is that's in fact more than all the numbers of genes in the human genome, encoded for by just one gene in the lowly Drosophila. Now you might be wondering, why the heck is this gene behaving this way? It turns out that DSCAM plays a critical role in forming neural complexity. Now, when axons are guided through the developing nervous system, there are different avenues that an axon might split into. So in this particular case, we have a blue axon and we have a gray axon. And this axon needs to split in this direction and this direction. Blue needs to go both into the dorsal lobe and into the medial lobe. Likewise, gray needs to go into the dorsal lobe and the medial lobe. Now, the way that these two parts of this blue exon will split apart from each other is through something called self-recognition and repulsion. So basically, blue, when it comes to this split, will repulse the other blue exon. The other blue axon. Right? It should be more clear. We're talking about axons. We're talking about cells here. We're not talking about exons at this moment. We will briefly, in a brief moment, shortly, but right at this moment we are. So these axons, all right, these cells, repulse each other, and so there's a recognition, and so blue will repulse blue, and you'll get a branching. But you wouldn't want blue to repel gray, because blue should grow in its own way, and gray should grow in its own way, and they shouldn't repel each other. So what this ends up happening under wild-type conditions, blue will branch and split in this particular way, and gray will branch and split in this particular way because blue is repelling blue, and gray is repelling gray, but blue isn't repelling gray, and gray isn't repelling blue. And so you get this proper branching, which is called arborization. And arborization, basically means tree-like structure. So under wild type, when DSCAM is wild type and has this ability to generate these different exons structures, it will show a normal branching structure in the nervous system. And the reason, let me make sure, the reason that these are able to do this is each of the cells, here the blue axon, right, expresses its own form of DSCAM based on that splicing reaction. So the blue one will generate one version of the DSCAM splice form. The gray will generate another version of the splice form. And so that means the splice forms themselves identify the signature aspect of the blue exon, and it's the blue exons which express a particular splice form have the ability to repel the other growing end that also expresses the same splice form. However, gray repels only gray because it only repels the other basic cells that express that particular version of the splice form. So because you can imagine that the nervous system has many cells and that have to interact with themselves and with other cells properly. And so these DSCAM splice variants basically mediate identity formation within the growing nervous system to enable proper arborization. In this mutant here, there's a defect, and the red is no longer able to properly repel itself because it's a DSCAM mutant. The DSCAM gene is missing, and because it doesn't properly repel in this sort of branching process, then you'll end up with defective neurons that look kind of like that. So the point here is that DSCAM plays a critical role in forming neural complexity that arises out of the fact that DSCAM can splice in all these different ways, and in each cell of the brain can adopt a unique identity based on a particular fingerprint that is established through the splicing reaction.